The next step in the process is to define cell types to assign individual cells to known cell lineage or functionality. MAS provides two methods to define cell types. The first, a method that is very commonly used in the cytometry community, is gating, and the second is clustering. Using gating, you can define cell types by the expression levels of well-established cell lineage markers. You can define cell types on the cell types page here. For example, we can create a plot of CD45 expression and CD14 of all the cells for a sample. In this plot, each point represents a cell. The blue color indicates cells that are sparsely distributed in a given region, while the red indicates many cells are densely distributed within the same region. We know the lymphocytes express CD45, therefore we can define lymphocytes using a rectangle gate and naming them lymphocytes. Similarly, we know that CD14 is expressed in monocytes. We can save the definition of monocytes and lymphocytes to all the samples by simply clicking on Save to All. Once we have captured lymphocytes, we can further gate these into T cells and B cells by using a T cell marker, for example CD3, and a B cell marker, for example CD19. Note that gating, in addition to capturing cells with specific expression patterns, also captures the hierarchy. Finally, here we will capture the next NK cells by adding another plot based on CD3 expression and an NK cell marker, for example CD56 expression. Under gating, there is additional functionality you might find convenient. For example, it's possible to copy cell types from a previously analyzed sample by simply using copy gating and selecting a sample that was previously analyzed. You can search here for any sample that's been previously analyzed in MAS. In addition, you can also display the plots either by sample, which is the default display here, all the plots belonging to a sample in one group, or you can change the plots to be displayed by the type of plot. So in this case, for example, you're seeing all the monocyte lymphocyte plots for all the samples together, so you can compare and contrast and make any adjustments necessary by simply clicking on a plot and changing it around. Once you change, you can either save it only to that sample or save to all of them. Finally, you can also clone a plot by clicking on this and quickly changing the input cell type, but keeping the axes the same. Unlike gating, clustering is a more automated method that is used to discover cell groups based on expression pattern. It is more appropriate when cell lineage is not clear. Note that clustering can be combined with gating. For visualization purposes, it is beneficial to have two-dimensional projections for UMAP and t-SNE defined prior to clustering. So before we proceed to clustering, let us switch to projections menu here and then come back. You can define as many projections as you like. You can start by clicking on the plus sign here. Then select the cells of interest. By default, this is all the cells that belong to a sample. But if you are focused on studying a specific cell subset, you can select one of those cell types, in our case, T cells. Then select the markers of interest. First of all, give it a description. Select the markers of interest using this menu. You can select either ADT or RNA, but we would not encourage selecting both simultaneously for the same projection. Like I mentioned before, it's possible to create multiple projections with different choices for markers, input cell types, algorithms and other parameters. The plot here, I'm going to unselect all the RNA for this example. Under ADT, the plot here is showing the range of expression for each marker. The blue lines here with different shades of blue give you the extremes of a specific marker, along with the 75 and 25 percentile levels, the 95 and 95 percentile levels, the 25 and 75 percentile levels, and the median expression all combined together. It is generally useful to, for example, filter out isotypes. Once you have selected the markers, click OK. Then the last step is to select the type of algorithm you want to run. You can click on additional options to set up other types of parameters for each of these methods. There is additional information about each of these projection methods captured under this menu. In this case, I'm going to select Disney, and you can click on Preview to run the algorithm. This will take about 15 to 30 seconds. A quick review of the results can be done right here, for example, by overlaying different markers on top of the Disney that was just computed. You can save this projection to the samples by clicking on Save. Once a projection has been created, one or multiple for that matter, we can now go back to the clustering page to create new cell types. We can do that by clicking on the cell types again, and then clicking on the clustering page. Here, we can click the plus sign to create a new cluster. Set up a description for the clustering. And as in the case of the projections, you can select the markers that are of interest for creating clusters. In this case, again, we're not going to use RNA, but it is possible to use the RNA dimensions as well for defining clustering. Just a note of caution here, not to use RNA and ADT in the same clustering exercise, simply because they are usually on different scales. Here again, I'm going to filter out the isotypes, click OK. That selected 153 ADT parameters that are input to the clustering, that will be input to this clustering algorithm. Now, our interest is actually in subdividing the T cells. So I'm going to select T cells as the input cell type. And we would recommend either selecting Park or Leiden as your choice of algorithms for clustering. Here again, additional options are available to set various parameters. A critical parameter for Leiden or Park algorithm is usually resolution, which defines how fine-grained or coarse your cluster should be. A value somewhere between 0.25 and 0.5 is recommended when using ADT data. Once I've selected the algorithm and various other parameters, I can click on Preview to run the clustering method. This may take a few minutes. The results for the clustering are shown in a bubble plot. Laid out across the rows are the different clusters that were defined by the algorithm, and a name that was auto-created by the method based on the various expression patterns that were observed, the markers that had the highest differential expression from other clusters. The third column here is the frequency, what fraction of the T cells that we used as input are captured in that cluster, and the variance that we observe across those cells, across the samples for that frequency. Then finally, on the columns are the markers that are the distinguishing features for each of the clusters. The top three markers that are differentially expressed, the color indicating more yellow, the higher the fold change of that marker above the rest of the clusters, and the size of the cluster or size of the bubble indicating what fraction of the cells in that cluster are considered positive for that marker expression. In the case of ADT, the positivity is computed against a background. In the case of RNA, it's simply what fraction of the cells have a non-zero expression. You can conduct additional tasks here. For example, you can decide that that is too small a cluster, and you can either delete that cluster, or if there are clusters that have very similar patterns, you can actually merge those clusters. For example, let's say I want to merge both of these clusters because they seem to have very similar patterns with very small differences. In addition, you can interact with this bubble plot by clicking on any of the rows, which should allow you to visualize the expression of these markers, actually the top 10 markers for that cluster, using the projection that we just created. If you had multiple projections created, this will provide a choice of which of those projections you want to use for visualization. These are the top 10 markers based on their differential expression against everything else. And you can see here the Disney colored by the clusters that were defined, all the clusters, and here only that subset of cells displayed. The remaining plots here, each one is colored by the top 10 markers for that cluster and their expression patterns within this cluster. Once you are satisfied with the cluster definitions, you can simply click on Save to save these cluster definitions to the samples. Now, if you go back to gating, you can actually see that T cells have been split based on the clustering. And this should indicate to you quickly that clustering and gating can be combined with each other. You can take gated cells and then subdivide them using clustering and vice versa. I should also note here that projections are not defined just for clustering. They're just useful when doing clustering, but projections are also used downstream in conducting various exploratory analysis, as you will see in the analysis step down below. Once the cell types have been defined, the last step in the process is to conduct various types of analysis that is covered in the next step.